And it's like that beginning never happened. All right. All the way to beginning. All right. So here we go. We're gonna go through kind of the beginning of the E2 material. Definitely get through the muscarinic, nicotinic drugs, and then we'll do beta alpha tomorrow. There's a lot of tips and tricks that I put in here for you guys. But obviously I don't know what specific drugs he's going to have you memorize. So just because I list drugs in this slide deck doesn't necessarily mean they are all the slides that or all the drugs that you have to know. So just keep that in mind. All right. So when we talk about the nervous system, where we always have to start with is understanding what kind of receptors we are dealing with. And this is what really lends itself to the biggest, I think, confusion point. Everything in the slides. Okay. The biggest confusion points in the second exam is where are all these receptors? And specifically, how do we know that the body is interacting with these different things? So in general, what we can say is that most tissues are going to have both muscarinic and adrenergic involvement. So what we're talking about here is that they have, oh, so awful, awful. What we're talking about is that they have both parasympathetic and sympathetic involvement. Can you guys see the annotations, by the way? Thank you,
Yes.
Jonah. Okay, sweet. So when we think of this, what I always want you to keep in mind is that parasympathetic is rest and digest, whereas sympathetic is fight or flight. So what we talk about really in the nervous system is the two different kinds of tones that the body uses. And when you are increasing the parasympathetic tone, or there's more parasympathetic stimulation, we call this vagal tone. And so what is going on in the body? Well, the body is able to digest, it's able to produce secretions, it's able to let out the bladder, produce feces, all the sorts of things that are associated with resting. Conversely, what we have is the sympathetic tone, which is everything that happens when the body is actively using energy. So a lot of this could be, it's not light exercise, it's heavy exercise. So you're still in vagal tone if you are walking up the stairs, but if you run up the stairs, then you switch into sympathetic tone. Likewise, if you're scared or if there is a surge of cortisol into the blood, then you have you're increasing the sympathetic tone. And these are opposites of each other. And so when we think of these tissues, what I want you to bear in mind is that we have four categories of drugs. We have muscarinic, adrenergic, central nicotinic drugs, and peripheral nicotinic drugs. It's important to understand this difference between central and peripheral, because peripheral nicotinic drugs like varenicline are only going to be able to affect, not varenicline, peripheral nicotinic drugs are only going to be able to affect the skeletal muscle. While drugs like that are able to move into the central compartment, So meaning that we're talking about inside the brain, inside the CNS, are preganglionic. So if you are a drug that is not able to penetrate the blood-brain barrier, you are not able to affect these preganglionic nicotinic receptors. And that was always something that really confused me when I was learning this, is how can we have nicotinic receptors here and nicotinic receptors here, but they're different somehow? And the reason is because these nicotinic receptors can only be penetrated by drugs that can cross the blood-brain barrier. Muscarinic drugs are only going to be affecting the postganglionic muscarinic receptors, whereas adrenergic drugs are going to be focusing on the postganglionic adrenergic beta and alpha receptors. So most of the drugs that we are going to talk about are acting peripherally, right? Everything that is postganglionic is peripheral. Very few drugs are able to actually get into the CNS to provide this preganglionic involvement, which is what accounts for this difference with the different kinds of nicotinic receptors. Any questions about that? Does anybody have any questions?
So you said the muscarinic drug only affect the post-ganglionic muscarinic receptors, right?
Correct. Yep.
Thank you.
No problem. All right. Am I here? All drawings. So let's look at the different kinds of drugs. Like we said, two different nervous systems, so we have two different sets of neurotransmitters to control them. When we have vagal, rest, and digest, the predominant receptors are going to be muscarinic, 1 through 5. What we're going to have is preganglionically, we're going to have acetylcholine, and postganglionically, we're going to have acetylcholine. What this means is that the endogenous chemical that is going to be controlling muscarinic receptors is acetylcholine. So the drugs that we are going to be using should in some way mimic acetylcholine. Conversely, when we think about the sympathetic nervous system, we're talking about the alpha and beta receptors, and they're going to be using acetylcholine preganglionically, and then epi and norepi postganglionically. So what do you have to know for this? Really, all you need to focus on is that we have acetylcholine is rest and digest, and epi and norepi is fight or flight, right? This should make sense because over in England, they call epinephrine adrenaline. And they call norepi noradrenaline. So this is just another reinforcement point that if you see epinephrine or norepinephrine, we are talking about getting the body ready to run or ready to do some sort of action. The somatic motor system, so we're talking skeletal muscle, doesn't use pre and post ganglionic neurons. It uses direct stimulation. So what it's doing is it's releasing the acetylcholine directly onto the neuromuscular junction. So if you go way back to biochem, you'll remember that you have the nerve comes in and that stimulates the release of calcium, which then the myosin head and I don't even remember it all. But essentially what it is that you have a direct stimulation of the nicotinic receptor, and that's going to cause the muscle contraction. Okay. So all in all, what you need to be aware of is that acetylcholine in most of the body, so outside of skeletal muscle, acetylcholine is going to do rest and digest, and epi and norepi is going to do fight or flight. Skeletal muscle is going to be all about control. Any questions about this? All right. So acetylcholine is used everywhere. It's used everywhere in the body, but and what's important to know is that we're not just releasing acetylcholine to float around and activate these receptors. The distance between the presynaptic neuron, the neuron that's releasing the acetylcholine, and the postsynaptic neuron, the one that is receiving the signal, is really, really, really small. We're talking about 10 micrometers. So 5 times the diameter of your hair, right? So this is a very, very small distance, and it's a very controlled release of acetylcholine. So don't get confused that acetylcholine is showing up in all these different places, because it's not floating around the body activating all these different things. It exists in one place only, and it's doing one action only. So you get super, super, super targeted transmission, and that accounts for a lot of the tight control that the body has over the different tissues. The flip side to this is when we talk about drugs, we're administering it by the stomach usually. So, or even if we're giving it IV, it's going generally into the blood. So it's going to go everywhere in the body, meaning that if it can go to a receptor, it will go there. That in order for a drug to activate these, like I said earlier, in order for a drug to activate these preganglionic receptors, it needs to be able to get into the brain. It needs to get into the CNS in order to penetrate the blood-brain barrier. So the key point here is that when you think of cholinergic drugs, when you think of drugs that are affecting the muscarinic receptors, don't think about central action. All right. This is a nicotinic receptor is not the same thing as a muscarinic receptor. They're two completely different things. All right. So even though they're both acetylcholine, they are two very, very different receptors. So that's my little spiel that I always give, because this was a big point that always confused me because it shows up everywhere. All right. This was just a chart for you guys. I think it just summarizes things very, very nicely, in case if you have any questions. All right. What is, yes. So when we talk about our first drugs, what we're going to talk about is drugs affecting neurotransmitter release. And it's very, very, very important to know the steps of neurotransmitter release, because this is a key way that we can control the amount of neurotransmitter that is released in the body. So the first step is that the action potential comes down. So something up above stimulated this nerve, which then causes the calcium channels to open. This influx of calcium is going to cause the vesicles holding the neurotransmitters to start to migrate towards the cell membrane. Once it docks to the cell membrane, which we'll talk about those proteins on the next slide, the neurotransmitter is released into the synapse, where it can then cause the response that is going on. This is the minimum number of steps that you need to memorize. So this is a perfectly valid 8-step diagram. So for exam 2, this is definitely something that you need to be able to write like the back of your hand. Very, 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 very important. Any questions about this slide? Also, am I going too quickly or am I good? Okay. All right. This is kind of intro stuff from the first class. Just as a reminder too about anatomy, what is very, very important to know is the SNAPs and VAMPs. SNAPs are proteins that exist on the membrane of the neuron, whereas VAMPs exist on the vesicle. I definitely remember a question about where do SNAPs and where do VAMPs exist. So V for vesicle and then N for neuron. So please, please, please just kind of get that one into your brain because that was something that I forgot. The other important structure to remember is vesicular acetylcholine transporter. And when we talk about acetylcholine transporters, this is different than the catecholamine transporter when we talk about drugs like tyrosine, metyrosine, or droxidopa, stuff like that. So just know that we have two different kinds of neurons. The first kind of neuron releases acetylcholine and uses VAT as a transporter, whereas the other kind of neuron is using VMAT, vesicular monoamine transporter. Specifically in adrenergic receptors, so ones that are releasing norepi and epinephrine, it's really the control of VMAT that is enabling the catecholamine release. And we'll talk more specifically about that in just a second. Any questions so far about these two different neurons? So we have a cholinergic neuron, and we have our adrenergic neuron.
So one releases acetylcholine, the other one releases norep and epi. Pretty
Exactly.
much.
Yep.
All right.
Yep.
Thank you.
All right. First drugs. Will vamps drink my blood? They're in your blood. They're already there. All right. Every neuron has a specific set of proteins that help the vesicle fuse to the membrane. Each neuron has, a different kind of SNAP and VAMP. They're all labeled with tons and tons of different things. Specifically in cholinergic neurons, they have the SNAP25, which is what's helping the acetylcholine vesicle dock to the dock on the oh, that goes away. Ooh, I like that. So the cholinergic neuron uses SNAP25 in order to ensure that the acetylcholine will release. Our first drug, Botox, specifically cleaves this protein and cuts it off of the membrane. So if you don't have this protein on the membrane, then the vesicle isn't able to dock. And so you're not able to cause that release. Now, we know that there are two different kinds of receptors that utilize acetylcholine, right? We know, based on what we looked at before, that we have the muscarinic receptors that use acetylcholine, and we also have nicotinic receptors that use acetylcholine. Specifically, Botox is targeting nicotinic neurons. So the neurons that would release acetylcholine onto skeletal muscle are the ones that Botox affects. So you're getting, this should say, whoops, that is wrong. Decrease oh, no, that's accurate. My bad. Whoops. So in one of the other kind of newer aspects of Botox is that we can inject it into areas that have high levels of smooth muscle, and we can start to impact the smooth muscle that would be utilizing the same protein. And I believe that's using one of these other like Botox neurotoxin, like C or F. I can't remember. But what essentially we have is both a decreased control of the skeletal muscle and then also slight decrease of parasympathetic input. So let's break this down. When we have things like blepharospasm and strabismus, what we're trying to do is we're trying to cut off the muscle from being able to pull the eyes or pull the eyelids. And what injecting Botox does is that it destroys the body's ability to control the muscles in that specific region. For excessive sweating, what we're doing is we're injecting Botox into sweat glands, not to turn off the gland, but to turn off the smooth muscle that squeezes the gland and releases the sweat. So one of the big things to understand about glands is that they have these little spouts coming off, and they're all surrounded by smooth muscle. And it's when you squeeze the smooth muscle that the secretion comes out. So in this case, we're talking about sweat glands. So you inject Botox, you kill essentially the smooth muscle control, and you prevent the sweating from happening. Same thing with the bladder. You guys have learned that the bladder is surrounded by the detrusor muscle. And so you can inject Botox into the detrusor muscle to prevent it from squeezing the bladder. Any questions about that? All right. We see that Botox is reversible after 3 months, which is actually pretty helpful because, you know, maybe things get better over time. And so we don't need to permanently cause this degradation. So we have Botox, mechanism of action is to degrade SNAP proteins. And my question for you is, how does Botox being administered by local injection fit in with its mechanism of action and its uses? Why does that make sense? Yeah, very good point. We don't want systemic release, right? If you just put Botox in the body, which is botulinum toxin, right? Then you would start to prevent muscles from controlling everything in the body. So you might stop cardiac muscle, you might stop the diaphragm, you might quite literally turn off everything, right? And it's only reversible after 3 months. So if somehow you can keep the patient alive for 3 months, you know, maybe that's great. How about with its mechanism of action? How does that make sense? How does local injection make sense there? Or its uses? Why is it these very, very specific? I'm not sure actually. It's a really, really, really big molecule. I know that. It's a big protein. But essentially, that we're using these in local places, right? We're using it on specific sweat glands, we're using it on specific muscles in the body. So that means that we're actively searching for specific SNAP proteins, right? So you wouldn't use it in an area that didn't have uh, applicable SNAP proteins. Um, Well, if you're giving it by local injection, then it could just spill into the bloodstream. So it could just do that. We only really worry about crossing membranes if you're doing absorption, not necessarily just direct injection. Good question, though. The flip side to this is what happens if we want to increase the release of a neurotransmitter. So for Botox, we are preventing acetylcholine release, but for droxidopa, it is acting as a substrate of dopa decarboxylase, which is enabling more norepinephrine production. So what is essentially happening with droxidopa is that we administer it specifically as a precursor molecule. Droxidopa is a precursor that we expect to get metabolized into dopamine, which then gets metabolized into norepi. Two different reasons why we use it. Both end up having the same kind of effect. Is the first is that it's going to increase stimulation on the heart. And the second is that it's going to cause the blood vessels to constrict, which is going to increase blood pressure overall. So my question for you is this a more vagal tone or more sympathetic tone? Which one are we increasing in the body? Vagal tone or sympathetic?
Would it be vagal since you prevent the actually yeah, it would be sympathetic, never mind. Yeah.
Very nice. So what is happening here is that we're giving droxidopa, which is going to increase the amount of norepi. And we know that whenever we talk about norepi, we're talking about the sympathetic nervous system. And so we're going to increase the sympathetic tone of the body. And this makes sense, right? What is going on here is that we're increasing heart rate and increasing blood pressure, which is something that we need for when we're running away or we're going off and doing some sort of strenuous activity. Really what this would get used for is only for orthostatic hypotension. So for someone whose blood pressure drops rapidly when they stand up or they sit up too quickly, this is where droxidopa comes in. We have other kind of better drugs for orthostatic hypotension, I think, but this is the only one that kind of acts in this way. So, and we talked about that. Great. Just a little bit of medchem, just to keep you guys comparing it. So when you look at the difference between droxidopa and dopamine, and we can see that it's a very, very similar structure, right? And what this tells us is that we need this catechol backbone, but really all we need is to have this NH2 moiety here, right? This seems to be very, very important in order for it to get metabolized. Whereas when you look at carbidopa, which inhibits the same receptor or the same enzyme, it seems like having this azide link is a inhibition as well as this kind of methyl group here. So just a little bit of med chem for you. You don't have to memorize this slide. This is not PQL. All right, VMAT inhibitors. So we know that, do I have it on the next page? No. When we look at the steps of how norepi is created, what ends up happening is that we start with tyrosine, which gets metabolized through tyrosine hydroxylase into dopamine, which then goes through dopa decarboxylase and becomes dopamine. Sorry, this is L-dopa. Dopamine then needs to be taken up into the vesicle, where then it can be put through dopamine beta hydroxylase. I think that's the name, where it is finally converted into norepi. So in order for it to get taken up into the vesicle, it needs to go through VMAT. What happens if VMAT is inhibited? What happens if this is inhibited?
No norepi.
Yeah, you don't create norepi, and what ends up happening to the dopamine?
It'll go down.
Well, specifically, it goes through monoamine oxidase, right? MAO, and gets degraded So if you inhibit VMAT, what is going to end up happening to all that dopamine the neuron is trying to create? It immediately gets destroyed. So in order to prevent dopamine from being destroyed by MAO, it needs to get through VMAT. So the way that you should be thinking about this is that VMAT inhibitors are going to prevent norepi release. So if you have decreased norepi, what's going to happen to the tone of the body? Do we have increased or decreased sympathetic tone?
Decreased.
Decreased. Which means that we're going to have decreased fight or flight. Right? So with that in mind, we can say that we're going to have less heart stimulation, which means that we're going to decrease the heart rate, decrease the blood pressure. We're going to have decreased contraction of the blood vessels, which means that they're going to increase in diameter, which will decrease blood pressure. And so the overall effect here is that we end up with lower blood pressure. So when would this get used? If you have high blood pressure, right? So we can use the VMAT inhibitors specifically to help with hypertension. Why? Because they're going to prevent norepi release. We have a few different kinds. Reserpine is the only irreversible one of the bunch, whereas valbenazine, tetrabenazine, deutetrabenazine, these guys are going to be reversible for the most part. I think they still have really, really long half-lives, but I can't remember. Any questions about these drugs? All right. If reserpine yes, VMAT takes dopamine into the vesicle. Yep. So it doesn't change the molecule at all. All it does is it hides it in the vesicle and prevents it from being degraded. Good question. So if reserpine is irreversible, what does that mean about the duration of action? What should we expect for reserpine?
Too long.
Super long, right? So what does the body have to do in order to reverse reserpine?
Make more VMAT?
Yeah, essentially it has to get rid of the old VMAT that has been destroyed, and it has to make all new vesicles, right? So reserpine is a great drug for VMAT inhibition because it relies on the body to have to go through these processes rather than just regular old pharmacokinetics of molecules bouncing into each other. So duration of action is much, much, much longer than these other drugs. Yeah. So because reserpine is irreversible, it's going to stick to VMAT and prevent it from ever working. So the only way that the body can get rid of reserpine is by destroying the old vesicle and making all new ones. So until we can get new vesicles in, VMAT is going to be inhibited. So you get a super, super, super long duration of action. The flip side to this is met-tyrosine. So once again, you're going to be getting less norepi release. So we're going to prevent the tyrosine from being converted into L-DOPA, which then prevents it from being converted into dopamine. And specifically, what we are using met-tyrosine for is pheochromocytoma, which, can anyone tell me what pheochromocytoma is? Any chance? This guy here. Totally fine. So pheochromocytoma is a tumor in the adrenal gland. And what this is going to do is, like any tumor, it's like the tissue on steroids. It is just going to continuously release neurotransmitters. And which neurotransmitters are specifically coming from this tumor? norepi, which through a specific enzyme that is in the adrenal gland, MPMT, it releases epi. So the tumor is releasing norepi and epi in huge supplies. So someone with pheochromocytoma is going to have major sympathetic tone. So their heart is always going to be racing. Their blood pressure is going to be super, super, super high, right? They're going to feel like they are under attack all the time. And so what giving tyrosine does is that we are preventing all of the precursor from being made, right? So you're not going to have excess levels of dopamine. You're not going to have excess levels of norepi or epi, right? So the only time that we really use met-tyrosine is when we're talking about pheochromocytoma, all right? So are you going to really use it in hypertension because, you know, the heart is going a little bit too quickly? No. This is a pretty systemic drug. It's going to affect a lot. Does that make sense? Anyone have questions about met-tyrosine? All right. All right. Let's get to a little bit of practice. So take a moment, see if you can identify which drug should be at which point in the diagram. What do we think? What's going to be at 1? Tyrosine hydroxylase.
Met-tyrosine, even though it's not like a tumor. No?
Yeah, so this is, you know, a general diagram. I just tried to throw as many things in there. So the only drug that we have learned that would inhibit tyrosine hydroxylase would be met-tyrosine. Very nice. How about for number 2, inhibiting VMAT? What are our VMAT inhibitors? Yeah. Reserpine, tetrabenazine, val tetrabenazine, deutetrabenazine. All those guys. Very nice. How about a drug that would be affecting the snaps and vamps on the neuron? What affects snaps and vamps? Exactly, Botox. Now, this neuron is specifically showing like norepi and catecholamines, which we know that Botox would not work on this neuron. So just bear in mind that this should be a cholinergic neuron for Botox specifically. And then how about this transition from L-DOPA to dopamine? What drug would be working there? Exactly, droxidopa. Remember that droxidopa is a substrate of dopa decarboxylase, right? So it fits into the enzyme that goes right here, and it creates more dopamine. So expect on the exam to have a diagram like this and to have, certain positions you have to fill in, because this is a pretty important way of understanding how drugs interact and talk to each other. Question for you. Would you ever give metyrosine and reserpine at the same time? Would you ever give metyrosine and reserpine at the same time? Someone says no. How come? Why would you not give them?
You said metyrosine and tyrosine, right?
Metyrosine and reserpine. So these two drugs right here.
Yeah. I mean, at this point, nothing in the cell will be working except for the calcium channels. And that's literally pretty much it.
Basically, yeah, the VMAT is preventing dopamine, right? It's stopping this transfer of dopamine into the vesicle. But metyrosine is working upstream from that, right? So are you even making any dopamine to go through VMAT? No. So does it really make sense for reserpine to be administered with metyrosine? Not really, because you're working in the same flow of molecules. Does that make sense? You're basically building a dam down the stream, and then you're building another dam up the stream. It doesn't make sense to build two. Any questions about that? So metyrosine and reserpine is not a good combo because you're inhibiting the same pathway. One is upstream of the other. Best combinations of drugs are ones that work on different pathways. All right, let's get into the fun stuff now. When we talk about muscarinic receptors, please, 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 please remember this. The odd receptors are GQ, 135 or GQ, and the even receptors are GI. Okay? When you think of, I'll get to this table in a second. When you think of okay. When you think of GQ, I want you to always think about contraction, smooth muscle contraction. So GQ receptors specifically cause smooth muscle to contract. GI, however, is controlling the function of the cell. So M2 and M4 are talking about function, whereas M1, 3, and 5 is talking about contraction. Here's a table that you can use to categorize the effect of the sympathetic nervous system and the parasympathetic nervous system, and what different receptors we have going on. And as we go through the different drugs, we'll come back to this table and we'll fill in the information as we need to. All right. So let's talk about the eye. In the eye, we have the pupil, which is what allows light to enter into the back to the retina, which allows us to see and interpret it into vision. In the presence of too much light, we use the circular muscles to contract the iris and reduce the amount of light that is hitting the retina. The way to think about the circular muscles, or when we talk about the pupil constricting, we call this miosis. And the way that I always remember it is that meiosis is like my eye has swollen shut. All right. So this is a great way of remembering meiosis versus mydriasis. The way to think about meiosis is kind of like the aperture of a camera, except the only thing that the circular muscles can do is close. So they don't actually, open back up in this manner, but they essentially close the pupil in this circular muscle fashion. When we talk about mydriasis, we are using the radial muscles to pull the iris, to pull the pupil apart, which is going to allow more light to enter the eye. And this is a really good graphic for seeing exactly how those muscles allow for the pulling to go on. So this is someone having bright light shining into their eye, and you can see how those radial muscles are tugging on the iris. Like I said, when we talk about anything doing with smooth muscle, we're specifically talking about M1, 3, and 5. M1, 3, and 5 are GQ receptors, which are going to associate with that contraction. What's important to know is that M1, 3, and 5 are only found on the circular muscles. Okay? So what this means is when we specifically look at how M1, 3, and 5 are working, they're only found in the circular muscle. So they have no function here. So if you activate M1, 3, and 5, we're going to cause contraction, right? So activating the muscarinic receptors in the eye is going to cause what? Meiosis or mydriasis?
Meiosis.
Meiosis.
Meiosis. So the only way that you can cause meiosis with muscarinic receptors is through activation. Can we activate a receptor, a muscarinic receptor, to cause mydriasis? No. There are no muscarinic receptors in the radial muscle to pull the pupil open. So if we want to cause mydriasis, we need to inhibit M1, 3, and 5. Any questions about that? Because this is a really big point about how we use these drugs.
So to constrict the pupil, we need to activate the receptors, and to dilate it we need to inhibit the receptors.
Exactly. Yep. Remember, M1, 3, and 5, when they are activated, they cause contraction. So in the activation column, I should never see something like smooth muscle relaxation, right? Because if you activate GQ, you have to cause contraction of some kind. All right, we'll come back to this slide later. Oop, that was the last one I got to. So that means we move on to hand drawing. Oop, now I need to move my notes over. There we go. All right. Where else do we find M1, 3, and 5? We find M1, 3, and 5 on every single, or I should say, most of the different kinds of glands we have in the body. M1, M3, and M5. So like I said earlier, when we have a gland, they are surrounded by smooth muscle, which when they squeeze causes the secretion to come out. So glands only release their product when the smooth muscle squeezes them. So on the surface of these smooth muscle receptors, we have these M1, 3, and 5 receptors. And when these receptors are activated, GQ, we cause smooth muscle contraction. Which is going to release in glandular squeezing. So what kind of glands do we expect to be squeezed by M1, 3, and 5? Who can tell me some glands? Yeah, salivary glands, right? One of the big ones that we, whoops, I need my chart. There it is. One of the big glands that we see being innervated by M1, 3, and 5 is salivary glands. So we can make a chart. So when you activate versus inhibit. So for salivary glands, what is going to happen if you activate M1, 3, and 5 on salivary glands?
You'll salivate more.
Yeah, right? We're going to increase the release of saliva. If you inhibit, you're going to decrease the amount of saliva, right? What is a condition that we would want to increase the amount of saliva for?
Dry mouth, xerostomia.
Yeah, exactly. So xerostomia is a condition where someone doesn't produce enough saliva. So we can give a muscarinic agonist in order to increase the amount of saliva that they're releasing. Another gland, more specifically the smooth muscle that surrounds it, is the detrusor muscle, which is the one that surrounds the bladder. So what happens if you activate the detrusor muscle?
You pee.
Yeah, what she
Yeah,
said.
you're gonna pee. Tons of pee. What about if you inhibit, you're gonna decrease the amount of pee, right? So what is a condition that someone who needs to, in which we would want a muscarinic agonist to make them pee more? What would be a condition that would be
Urine
useful?
retention.
Yeah, urinary retention. Would there be a condition ever that we want to decrease the amount of pee? Overactive bladder. Very, very nice. And just so that way you guys have it very, very clear for you, here we are talking about muscarinic agonists, and here we are talking about antagonists. All right. Another one that we can have is the smooth muscle of the intestines. What would be the effect of contracting the smooth muscle around the intestines?
Constipation.
So diarrhea.
Yeah, so we're going to increase motility, right?
Right. Yeah.
We're going to cause the contents of the intestines to go further and further and further along, right? The other thing this ends up doing is it relaxes. I'm not going to talk about it. It's going to be too confusing. Doing an antagonist would decrease motility, right? So you guys already said it, but what would be a adverse reaction to a muscarinic antagonist? Diarrhea, right? The the um the feces are moving too quickly, right? Versus if you decrease motility, this is where you would get your constipation. Now, is there ever a condition where we would want to cause diarrhea? Not really. Is there ever a condition where we would want to cause constipation? Not really, unless if you have diarrhea, right? So we have better drugs that work more specifically on the colon and the intestines, because don't forget, right? If you gave this drug, it's going to affect your saliva, it's going to affect everything else. There is one indication that you might want to use it for its effect on the smooth muscle here, and it's called GI atony. And atony is where the essentially the smooth muscle is not moving anymore. It's basically turned off. And so by giving a muscarinic agonist, you can cause it to start to move, and you're going to increase the amount of digestion, because otherwise that food is just going to sit there and essentially rot inside of you, which is no good. No good at all. Let's talk about nasal glands. So specifically ones that produce mucus. What's going to happen if you give a muscarinic agonist here? Atony, A-T-O-N-Y. Go ahead, Pierre.
I was gonna say a runny nose.
Yeah, increase mucus. So this could produce a adverse reaction like a runny nose, maybe even a stuffy nose. No word unlocked. And if you gave an antagonist, we would decrease the amount of mucus being squeezed out of the nasal glands. Which one do you think is more useful? The agonist or the antagonist? Yeah,
Antagonist.
antagonist, right? And so which indication do you think would be used here? Yeah, stuffy nose, I would say. It would be used for a stuffy nose because you're going to reduce secretions. Stuffy nose, runny nose, that sort of thing. Would it cause a bloody nose if it affects blood pressure in the nose? Good question. Generally, we say that bloody nose is caused by generally a bloody nose is more about like dryness in the nose. So it could cause a bloody nose not because of blood pressure, but because of it makes the nose too dry and then it cracks and then you essentially have a wound in the nose, causing a bloody nose. So like when it's, really cold outside sort of thing. Yeah, then you need saline. Good question though. All right. So M1, 3, and 5, tons of effect on glands, the GI tract, and tons of effect on the eye, right? As we talked about earlier, that the meiosis versus mydriasis. So you can add this in on that table that I showed you earlier. Don't forget muscarinic receptors. What kind of nervous system is this? Parasympathetic or sympathetic?
Sympathetic? Oh, my bad.
Parasympathetic, right? Parasympathetic is rest and digest. Which column represents rest and digest?
The activate column or the agonist column, I guess.
Yeah, the activation, right? We're activating muscarinic, which means that we are causing rest and digest, right? Which means that we are increasing the vagal tone. One important point that I just want to get across is that obviously inhibiting the muscarinic receptors is going to inhibit rest and digest. Can you say that it causes sympathetic stimulation? Can you say that we're causing fight or flight? Exactly. No, right? That we can't say that we're causing fight or flight because what is the only nervous system that innervates fight or flight? Sympathetic, right? All we can do here is increase or decrease rest and digest. So it is not accurate to say that muscarinic antagonists cause fight or flight. They don't do that. They just decreased rest and digest. Does that make sense? So if you're talking about parasympathetic system, you can only talk about rest and digest. If you're talking about sympathetic, you can only talk about fight or flight. They look like each other, right? If you decrease rest and digest, you're going to have a lot of the same symptoms and cause and effects, but they're two different nervous systems. Okay. So even though that they look like they're causing the same thing, they are not going to work in that way. Any questions? All right. Sweet. Let's talk about M2 and M4. What color did I use? Blue. M2 and M4. Whoops. Oh, I was thinking of adrenergic. M2 and M4, the only places that you really find these guys is in the heart and the lungs. Can you find M1, 3, and 5 in the heart and lungs? Yes. In fact, we find a lot of M3, but in very, very small amounts compared to M2 and M4. So if you have a question about what the predominant receptor is, a muscarinic receptor in the lung or the heart is, M2 and M4. All right. So M1, 3, and 5 is everywhere else in the body. M2, M4, heart and lungs. Remember that M2 and M4 are GI-linked GPCRs, which means that we're going to be decreasing the amount of CAMP being produced. Let me see if I can get a picture. No. All right. Normally, what ends up happening in the lung is normal function. In the lung, acetylcholine would activate no, how do I want to? Yes. Acetylcholine is going to cause the M2, M4 receptors to activate. What this is going to do is twofold. You are going to cause the Oh, what's the best way of explaining this? I had a really good picture, but now I can't find it. One moment. Ah, okay. I found it. I knew I could do it. All right. Normally, when we talk about the lung tissue, what we have going on is we have nice lung, and we have the GS receptor. And we have the GI receptor. Oop. GI and GS. So in the lung, it is innervated by both GS and GI. Lung tissue. The first thing I want to talk about is GS, where once GS is activated, it is going to affect adenylate cyclase, which is going to convert ATP into CAMP. We know that CAMP will then go and activate phosphokinase A. And specifically what is going to happen here is that we are going to affect calmodulin, which is going to cause bronchial relaxation. So the effect of GS is that the diameter of the bronchiole increases. What would happen to your breathing? Would your breathing get easier or harder once GS is stimulated?
Easier.
Easier, right? Because the diameter of the airway is much, much larger. What's going to happen with GI is we know that GI inhibits adenylate cyclase, right? So you're going to end up decreasing bronchial relaxation, which is going to end up looking like constriction. Okay. M2 and M4 are the GI linked receptors. And so in the lung, the muscarinic receptors inhibit relaxation. What does this appear like? Constriction. So if you give a muscarinic agonist, what is going to happen to your breathing? Is it going to get better or worse? If you give a muscarinic agonist,
Worse.
Worse.
it's going to get worse. Why? Because you are going to inhibit relaxation. If you're wondering what receptor is GS, does anybody remember what GS is? Which
The betas.
it is beta 2. So beta receptors are going to, when you agonize the beta receptors, you cause relaxation. But if you agonize the muscarinic receptors, you're going to cause constriction, right? So when we look at our chart, I don't have any room. When we look at our chart and we talk about activation versus inhibition, in the lung, activating M2, M4 is going to cause bronchial constriction. Whereas inhibiting the muscarinic receptor is going to cause bronchial relaxation. Is there a condition where we would ever want to relax the bronchioles?
Asthma.
Asthma, COPD, emphysema, all your airway diseases. Is there ever a condition where we want to constrict the bronchioles? There is one. There's one. If we want to kill the patient. So is a muscarinic agonist a good idea for someone who has severe asthma? You're allowed 4 a year, Sarah. You can kill 4 people a year. Muscarinic agonists are not a good idea for severe asthmatics, severe COPD. Not a good idea. Any questions about this? All right. Still talking about M2, M4. But now specifically, we're going to look at the heart. In when we look at the heart, actually, I think it's just the SA node. Heart, SA node. Same thing goes on previously. All right. We have GS. And we have GI. Oh, you included the myocytes. Okay. I thought the myocytes were involved, but I'm not looking at the chart. You should be looking at the chart. Yes. Yes, yes, yes. So what happens in the SA node in the myocyte? Okay. Just like before, GS is going to stimulate PKA, which is going to increase CAMP production, which is going to cause two effects. One, oh, sorry, not PKA. Adenylate cyclase. One is our good old friend PKA, which in this case, what is going to happen here is that calcium channels on the surface of the heart cell are going to be opened, which is going to stimulate contraction. Right? We know calcium is a major factor in the amount in contraction. The other part to this is oh no, just one part. So GS on the heart cells is going to increase contraction. I believe there's also going to be some factor that has to do with, I'm sure, hyperpolarization. I just can't remember. But for the most part, this is a big aspect. The GI receptor in this case, once again, it's going to inhibit adenylate cyclase, which is going to decrease the amount of CAMP, which will decrease contraction. So our muscarinic agonists, the first effect that they're going to have is they're going to decrease CAMP, which will decrease contraction. The other effect that the muscarinic receptor has is that it opens potassium channels allowing for potassium to escape the cell and hyperpolarizing it. So the first effect is we decrease CAMP. The second effect is we cause potassium efflux, which will hyperpolarize the cell. And what is the effect of hyperpolarizing the cell? What is that going to do to the heart?
Activity decreases.
Which specific activity? What measurement would it be?
Would it be a SA node firing?
Exactly. Yep. So we're going to decrease the heart rate, right? We're going to cut down, we're going to make it harder for the SA node to fire, which means that it's going to take a much bigger stimulation for the SA node to make a beat happen. Any questions about these two effects of a muscarinic agonist? All right. So, by the way, what receptor is the GS? Can anyone tell me?
Beta 1?
It is beta 1. And we'll talk about beta later on, probably tomorrow. But these are the beta receptors. So finally, when we talk about the muscarinic drugs, when we talk about the heart, when we activate M2 and M4, activating M2 and M4 on the heart is going to cause decreased heart rate, decreased conduction velocity, and a decreased contractility. Exactly. Yep. Yep. If you inactivate, you have the opposite effects. Increased heart rate, increased conduction velocity, and increased contractility. Once again, rest and digest, less rest and digest. What indications would we want to use the muscarinic agonist for? Yeah, fast heartbeat, heart failure. The only thing is that we don't really use muscarinic agonists like this. Why is that? Why do we not use muscarinic agonists for their effects on the heart?
Side effects?
Tons of side effects. So back in the day, when we only really had muscarinic agonists to use, yeah, we would use them for hypertension, we would use them for heart failure, that sort of thing. Nowadays that we have better drugs, we don't really use them on the for the effect on the heart. Okay. So what I want you to know is that for the heart, the important thing to know is that the effect on the heart are side effects. There are no indications to use a muscarinic agonist or antagonist specifically for the heart. It's just not useful. So if you're going to be using a muscarinic agonist or antagonist, and we're talking about the heart, we're only talking about them in terms of God, what am I saying? In terms of side effects, that sort of thing. Yeah. All right. What I want you guys to do is go back to this chart that I gave you and fill in the information so that way you can all have it in one. And just know that when we're talking about activation, we're talking about agonists, and we're talking about inactivation, we're talking about antagonists. The big thing to know about the drugs for the exam is about the entire drug profile. Okay. Here we go. So when we talk about giving a drug, and we talk about systemic effects, right? For example, if I was to give carbachol, which is a muscarinic agonist, which receptors is carbachol affecting? Which receptors is carbachol going to affect?
Yeah, all the M1, 2, 3, 4, and 5.
Exactly. Carbachol is going to affect all of the muscarinic receptors. Even though we group them evens and odds, the agonists don't care. They go to all of them. We don't have specific drugs that only go to one receptor. All right. So what that means is that you're going to end up with the side effect profile. You end up with the effect profile of M1, 3, and 5, and the effect profile of M2 and 4. All right. So when you think about these muscarinic drugs, please keep this fact in mind. All right. You activate all the receptors, even though we split them up as evens and odds, so it's easier to talk about. That does not mean that you were targeting specific receptors. Okay. If you talk about a specific tissue, then that's a different matter. Okay. Any questions about the muscarinic drugs? All right. All right. I want to move on and talk about the last drug for tonight, cholinesterase. In the periphery, we know that we have two different kinds of neurons that release acetylcholine, right? So the first kind of neuron that takes acetylcholine and dumps it into the synapse is what kind of neuron? We just talked all about them. What kind of receptor would we expect here?
The M?
Yeah, the muscarinic receptors, right? So muscarinic neurons utilize acetylcholine endogenously, right? What is the other kind of receptor? The other kind of neuron that also releases acetylcholine, but are not muscarinic receptors. They're different receptors.
Nicotinic.
Exactly. Nicotinic. Okay. Both of these different kinds of receptors, whether we're talking about nicotinic or we're talking about muscarinic, use acetylcholine endogenously, right? And that was that whole spiel we talked about, how acetylcholine is everywhere in the body, but it's used very locally. We just talked about all the different tissues that muscarinic is used on, but what is the only kind of tissue that we're going to find nicotinic on peripherally? Where are we going to find peripheral nicotinic?
Skeletal muscle?
Yeah, exactly. Skeletal muscle. So, in the body, we have a enzyme in the synapse called acetylcholinesterase, ACHE. And what acetylcholinesterase does is that it takes acetylcholine, whether we're talking about a nicotinic receptor or we're talking about a muscarinic receptor, and degrades it into inactive metabolites, into choline and acetate, I think. All right. So if you give an acetylcholinesterase inhibitor, first point, an acetylcholinesterase inhibitor affects muscarinic and nicotinic receptors. If you give a acetylcholinesterase inhibitor, so an acetylcholinesterase inhibitor will come in and turn off acetylcholine. Will turn off the esterase. What's going to happen to the amount of acetylcholine in the synapse? What happens to the amount of acetylcholine in the synapse?
It will increase.
Increase. So what does that mean about our muscarinic activation? Are we going to get more or less muscarinic activation?
It will be overstimulated.
Tons of muscarinic activation, right? So you're going to cause the heart to slow down. You're going to cause the bronchioles to constrict. You're going to have a ton of effects. You're also going to increase nicotinic stimulation. And this is really what we use the acetylcholinesterase inhibitors for, is the effect on the nicotinic skeletal receptors. All right. So sorry, give me 1 second. There we go. So for the nicotinic receptors that are on the skeletal muscle, what we end up doing is we have what's called depolarizing depolarizing and non-depolarizing. Oh. Yes. For our acetylcholinesterase inhibitors, we have two different, the major component of them is that they are going to inhibit acetylcholinesterase at the skeletal muscle. Yes, I can do that. What this is going to do is that we're going to activate all skeletal muscle at basically the same time. Right? And so what did you guys learn earlier that the purpose of doing this is? If we're going to be activating all these different, exactly, paralysis. The purpose here is paralysis. The purpose of doing something like this is in a emergency situation where it wouldn't be good to have the patient moving around or to have the muscles tensing. So, for example, we're going to be intubating the patient. So we're going to be putting the tube down their throat in order to do mechanical ventilation. What specifically are we using the paralysis for? Why do we need to paralyze the patient in order to do this? So they don't asphyxiate and reject it. Why would they reject it, is my question. Exactly. We need to relax the airways. So it's not necessarily, some people have the idea that the paralysis is to like keep the patient from fighting you. That's what it's for. What the purpose of the paralysis is to relax the muscles to allow for us to do certain procedures. The other aspect that we use it for is in surgery, like if you're doing abdominal surgery. What would be the purpose of using it in surgery? Exactly. Relax muscle for cutting. Would you use this to sedate a patient? No, because it's not gonna sedate them. It's just gonna you're gonna paralyze them. And so they're still gonna be awake, but they can't do anything about it. And then their diaphragm is paralyzed, so they're gonna suffocate. So you only want to do this in a way where you're immediately going to intubate the patient because they're going to stop breathing. Not great. When we look at the duration of the acetylcholinesterase inhibitors, for example, we have so for duration of action, pancuronium has a slow onset, slow duration. Whereas pipercoronium has a fast onset, slow duration. Which drug is going to be which drug is going to be better for intubating the patient? The pipcuronium. How come? Why? Exactly. If we want to intubate the patient, we want this to work very, very, very quickly. We want this to happen within seconds, not necessarily minutes, right? A slow onset means that we're going to be waiting and waiting and waiting and waiting, which is not beneficial for anyone. So a faster onset is preferred. Do we want a slow duration or a fast duration? Slow duration or a fast duration?
Slow.
We would want to slow, right? The reason is because it's going to last longer, right? Now, Sarah makes a good point, right? That we're going to paralyze the diaphragm, but we can get around that by intubating the patient, right? We know we're going to have to intubate them anyways. So usually we're just going to be breathing for them. And until the drug wears off, that would be what we use it for. Same thing with surgery. You'd intubate them for the duration of the surgery. Yeah. And afterwards until they're breathing on their own. Any questions about this sort of stuff? All right. The other one that we need to talk about is succinylcholine. Succinylcholine is an analog of acetylcholine. Essentially what this does is it directly activates the nicotinic acetylcholine receptors. So instead of raising whatever levels of acetylcholine are already there, we're actively binding to it and turning it on. What this is going to do is we still get paralysis. All right, so we're still going to get the relaxing of the airways, relaxing of the muscles for surgery, that whole business. The difference here is that this is what's called a depolarizing neuromuscular blocker. Because it's directly binding to the receptor as acetylcholine would, it is going to cause a major, just looking for my notes. Where are you? There we go. It's going to cause an immediate reaction with the muscles in the body. One of the effects that happens with succinylcholine is that you end up getting a major energy release from all of that muscle seizing, which ends up getting producing a malignant hyperthermia. So we're talking about temperatures above 106 degrees Fahrenheit. Very bad. So in for people who overdose or where they're especially sensitive to succinylcholine, we might have to put them in an ice bath or cool them down in order to prevent them from literally cooking alive. That sort of thing. Yeah, not great. Not great at all. All right, this is where we will end with today. Let me go back to the lung real quick. Nope, that's the heart. Tomorrow we're gonna finish up the last little bits of the muscarinic and nicotinic drugs that we have to finish up. I'm going to specifically talk about myasthenia gravis, just because that is something that always confuses people. And then we'll jump right into beta and alpha and all that good stuff. And we'll get that all done. Great. And we should be good.